Hello everyone, and thank you for attending today's webinar, Bioanalysis by Hybridization ELISA to support recent advances in the field of antisense oligonucleotides. I'm Alex Hyde, editor of Bioanalysis, and I'll be your host for today's event. Before I introduce you to our speaker, I'd like to quickly cover a few housekeeping items. Firstly, all of the widgets that you can see on your screen are resizable and movable, so do feel free to move them around to get the most out of your desktop space. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right-hand corner. Please let us know your thoughts on today's webinar. You can tweet us at Bioanalysis using the hashtag BZWebinars, or simply share today's webinar with your friends and colleagues using the Share This widget that's on your screen. I'd just like to let you know that some networks can cause slides to advance more slowly than others, so logging off your VPN is recommended. And if your slides are behind, if you push F5 on your keyboard, you can refresh the page. You can find additional answers to some more common technical issues located in the Help widget that you can find at the bottom of your screen. And in terms of an on-demand version of today's webinar, that will be available approximately 1 hour after the webinar is completed, and it can be accessed using the same audience link that was sent to you earlier that you are accessing this webinar on. So we are delighted to have Danielle Salha from Altasciences presenting for us today. Dr. Salha joined Altasciences in 2017. As Director of the Ligand Binding Assay Department, Dr. Salha leads a team dedicated to bioanalysis to support preclinical and clinical PK, PD, and immunogenicity studies. Prior to joining Altasciences, Dr. Salha worked at Charles River Laboratories, where she acted as a study director to support immunogenicity studies. Previously, she worked at 14 years for Sanofi Pasteur, where she led the development and validation of potency assays to support clinical phase 1 and 2 product release. Dr. Salha received her PhD at McGill University from the Department of Immunology and Microbiology. Thank you very much for joining us today, Danielle. We do invite the audience to submit any questions you may have for our presenter throughout the webinar using the Q&A widget that you can find on the right-hand side of your screen. We will pose your questions to Danielle in the live Q&A session after the presentation, and then we'll follow up any outstanding questions offline. So thank you again for joining us today, everyone, and I will now hand over to our speaker, Danielle, to start today's presentation.
Thank you, Alex, for this introduction. So for today's webinar, I will be talking to you about the bioanalysis by hybridization ELISA to support recent advances in the field of antisense oligonucleotides. So in my presentation, I will be discussing first some background information regarding oligonucleotides and the different categories. Then I will move on to the bioanalysis of ASOs, which are antisense oligonucleotides, and the type of methods that we use to quantify them. Then I will discuss 3 case studies that illustrates the hybridization ELISAs and some troubleshooting that we've done to each of them. And then we'll finally finish off with a Q&A session. So oligonucleotides are usually small sequences that are complementary to the target mRNA sequence, and they comprise between 15 to 20 nucleotides. Typically, those oligonucleotides, if they're unmodified, they would be rapidly degraded by nucleases, and therefore it's highly recommended to chemically modify the ribonucleotides so that they're protected from the degradation by nucleases once injected into animals or humans. The chemical modification will improve the target affinity and delivery into the target tissue or organ. Another approach that has been taken by several companies is to covalently attach the oligonucleotides to various vehicles that will improve their biodistribution into the cell as well into the tissues. And those could include ligands such as peptides, proteins, carbohydrates, etc. There are different types of oligonucleotides for different ranges of applications. So the one that is mostly looked at and developed are the antisense oligonucleotides. These are inhibitors of RNA activities. They're usually short in length. They can be either single-stranded or double-stranded in the case of siRNA. And the role of these ASOs is typically to reduce or restore protein expression through several distinct mechanisms. We can also consider aptamers. So aptamers are also single-stranded oligonucleotides that fold into defined architectures, and those can bind to targets such as proteins. There's also mRNA that typically would encode for therapeutic proteins, and the purpose of those would be for vaccine administration, and so therefore boosting immune responses against therapeutic protein in question. There are also CPG, immunostimulatory oligonucleotides. So these are adjuvant-like oligonucleotides that would be conjugated together with some proteins in question for vaccine purposes to boost your immune response. And finally, we have the genetic information reprogramming nucleic acids, which are also known as CRISPR. And those are also very well known in the field, and recently there's a lot of research that has been developed with those CRISPR. The main purpose of those is to inhibit specific types of gene expression. So oligonucleotides have been around for many years, and the purpose of this slide is to just show you how many different discoveries have been made throughout the years. In pink, you see recently how oligonucleotides have entered into phase 1 or phase 2 clinical trials, and how they really have started to be used. And the bottom line is we started using oligonucleotides effectively when we started to identify means to increase their half-life by linking them to different types of ligands that has increased their half-life, or by providing the chemical modifications that allowed them to be effective. So what are the different regulatory considerations for oligonucleotides? Well, first of all, we follow small molecules because they are synthetic analytes. And there's no ICH or FDA regulatory guidelines that specifically address the expectations or standards for oligonucleotide products. We know that, as I showed you before in the previous slides, that there are different types of oligos, such as the aptamers, the ASOs, the mRNA. So there are different types and different mechanisms of actions, and what that leads to is that there are different diverse toxicology profiles or concerns that one needs to consider when developing oligonucleotides for therapeutic and human use. Oligonucleotide-based therapies, therapeutic toxicities can be due to, for example, interactions between the oligonucleotide and different types of molecules. For example, the Watson-Crick base pairing to intended nucleic acids or independent mechanisms. And also ASOs can bind to different proteins, such as human serum albumin. And this interaction can then serve as a carrier to induce now immune responses to the oligonucleotides, when in fact we know that the ASOs are not immunogenic, in fact. But the increase in half-life will provide and the binding to different proteins will play hand in hand and increase any level of immunogenicity against the linkers, against maybe even the oligonucleotides itself. So something definitely to consider when working with oligonucleotides. So now I will talk to you about the bioanalysis of oligonucleotides. What are the different strategies that are used by the industry to quantify oligonucleotides for the purpose of supporting pharmacokinetic studies? Well, there are several methods or approaches that can be utilized to quantify oligonucleotides. The first one that I will be more discussing in my presentation today is the Hybridization ELISA, or ECL. There's also the LC-MS-MS-based approach. There's the LC-FD, fluorescent detection methodologies, as well as PCRs. So if we are to look at the method-specific parameters for each of the approaches that I've described to you earlier, we can consider that, for example, for the hybridization assay, sample preparation is usually not required. Throughput is we can automate it very easily, and hybridization ELISA can have very high level of sensitivity. In terms of interference and specificity, the method is highly specific to the parent oligonucleotide. However, we may have some cross-reactivity with N1 and N-1 and N-2 metabolites. So definitely something that needs to be assessed during method development or validation. I will be describing how some methodologies are more prone to seeing these types of cross-reactivity, while others may have reduced level of those types of cross-reactivities. Reagent availability is required, obviously. We need to custom design the probes that are specific for the oligo in question, and the assay itself follows the LBA guidelines for validation. In terms of PCR and their requirements, sample preparation is usually required. Throughput is between medium to high. The sensitivity is actually very good. However, in terms of interferences and specificity, the assay itself may be prone to some contaminations. Probes are required, and finally, there's no current guidance that are described for the PCR itself in terms of using this methodologies. In terms of the mass spec approach, oligonucleotide sample preparation is required. Throughput level, I would say it would be medium, and sensitivity, it could be potentially acceptable. Interferences is usually very good because it can quantify all metabolites, including the parent oligonucleotide. Reagent availability, depending on the assay format itself, we may have to generate some common reagents and specific probes. And of course, for validation, we would follow the LC-MS-MS guidance. So what are the important bioanalytical parameters to consider for PK profiles when quantifying oligonucleotides? Well, one of the first and most important parameter to ensure, at least in our experience, is the level of sensitivity that is required for the assay. And this level of sensitivity is required for all the matrices used. The tissue, and why is that? Well, first of all, the tissue concentrations that we determined in our assay will eventually impact the decisions that are made when moving from preclinical to clinical studies, and therefore impacting the clinical dosing strategies. The ability to quantify oligonucleotides to very low concentrations may have an impact actually on the calculations of this half-life. And so the more accurate and the more sensitive the assay is, the better readout we have on this oligonucleotide half-life. And finally, the later time points expected are usually have lower concentrations in various tissues, and therefore the higher sensitivity the method that we're using has, the better accurate reading concentrations we have at the last time points that we collect from the different tissues of various studies. Also, the lack of interference is also something that is very important for metabolites, and that's something that will impact, if it is the case, will impact the interpretation of the PK profile. And then finally, multiple approaches, in fact, are required, because each method does have her limitations and her advantages, and therefore to have a full PK characterization, it's better to have different types of methods that are utilized for different types of purposes. For example, if the metabolites identification is required, well, it's better to use the mass spec approach. If you need a very sensitive method and let's say you've identified the metabolites, but now you're really trying to build your PK profile and get those concentrations and, identify what would be the next stage, what would be the next dosing strategy that you will use, maybe hybridization ELISA is better because it has a higher throughput to analyze many samples that you might have. So really, depending on what question you have, the methods have to be carefully considered. So moving on to hybridization ELISA. So hybridization ELISA has been used by us and by others to support preclinical and clinical programs. They've used them to support programs such as toxicokinetic studies, PK studies, distribution and elimination studies. The hybridization ELISA can quantify oligonucleotides of various compositions and lengths. So we have used them, as well as others, to quantify RNA and DNA between 16 and 24 nucleic acids in length. There are three different varieties of the hybridization ELISA. We can consider the dual hybridization, the hybridization ligation assay, and the nuclease-dependent hybridization ELISA. And typically, we and others follow the FDA guidance and Crystal City recommendation, the 2018 FDA guidance and Crystal City recommendation to perform our validations, which are also typical of any large molecule LBA assays. So the first assay format is the dual hybridization ELISA. This format has an enhanced sensitivity when compared to the ligation hybridization ELISA. It also has a wider dynamic range. However, the disadvantage is that this assay format may have a cross-reactivity with 3' or 5' metabolites. The way the assay format is designed is that it has a short capture probe, which is typically biotinylated and binding to the streptavidin-coated plates, which will hybridize with the oligonucleotide in question, followed then by a detection probe, which is labeled with the deoxygen. So the major difference between the dual hybridization and the ligation hybridization assay is that the later does utilize a T4 DNA ligase, which will allow the ligation of the detection probe with the 3' end of the oligonucleotide in question. And so, therefore, non-ligated probes will be washed away. This will allow the shorter metabolites, 3' metabolites, not to bind to the detection probe and therefore enrich parental oligonucleotide quantitation. And then finally, nuclease-dependent hybridization ELISA utilizes the S1 nuclease, which is used to cleave anything that is single-stranded. So in this case, the difference, as you can see from this figure, is that the capture and detection probe is one, where on one side the capture probe is linked to a biotin, and on the other side it's linked to the label, which is the dihydroxygens. And so after hybridization, the complex is a double-stranded RNA or DNA, and then the S1 nuclease is added, which will cleave anything that is single-stranded. So now that I've presented to you the different assay formats for hybridization ELISA, I'd like to discuss a little bit the points that are important to consider when developing an assay. The first point is really around the need to increase the sensitivity of our assays. And this is really due to the fact that there's been a huge amount of advancement in drug delivery and drug design, which makes the products that we test and we quantify much more potent. We need less number of doses that are required. The doses are usually of lower amounts and therefore require more sensitive assays. And so the way we felt would be more appropriate and allows us to really increase the sensitivity of our assays is to shift from ELISA to ECL approach. Assay development and optimization needs to reduce the background noise, of course, which comes hand in hand with having a higher sensitivity and ensure a good separation from blank matrix. This comes by really designing well the probes, optimizing the conditions of temperatures and duration of each step of the method development. So the second point I would like to discuss is the importance of determining whether the method itself is selective for the parent compound or if metabolites potentially can quantitate. To do so, we typically recommend to test different lengths of metabolites during validation. And it is recognized by us and by others that longer metabolites such as N-1 and N-2 are likely to quantitate. There could be some approaches to utilize in order to limit the cross-reactivity with those metabolites in terms of having appropriate probe design strategies or the addition of S1 nucleases. And then finally, another important point to consider is the interference coming from the anti-drug antibodies. Anti-drug antibodies may interfere with accurate oligonucleotide quantitation, and that's because the antibodies and the oligos will form complexes, and therefore they would not be detected by the hybridization assays. However, there are strategies to mitigate this interference. One of them is the addition of proteinase K into the method, and second is the high level of temperature treatments that is used. Potential interference of ADAs can be quantified and is recommended to be quantified during validation by evaluating the presence or absence of ADA at different QC levels. So I would like to talk to you next about some challenges that we've encountered in Altasciences around quantitation of oligonucleotides in tissues. And the first example I'd like to discuss is tissue distribution. So as some of you may know, oligonucleotides are not evenly distributed in a given tissue, and therefore oligonucleotide concentration determination may differ if one is to sample at different sites. So in order to ensure successful ISR reproducibility, the whole sample tissue homogenate is highly recommended. And this is really to ensure that the homogeneity between sample extractions. So given that it's very important to take into consideration the sample size and volume early on and communicate properly to the clinical or preclinical sites the requirements for those samples that you're trying to quantify the oligonucleotides. We did have an example in which we have decided to go with a specific type of bags to collect tissue samples, and those bags were not appropriate for the type of homogenization because we intended to homogenize the whole tissue itself, and therefore instructions were provided to the clinical site to have bigger bags so that we have the ability to properly homogenize the samples. And once those were provided and everything went very smoothly, and that's why I thought this was a good example to consider quite early in the process of developing these methods, to really think about how those tissues are being collected and how the way they're being shipped to you for analysis is being done, and what would be those requirements. So as a second example, I'd like to discuss the tissue diversity and characteristics and how those may impact quantitation of the oligonucleotide in question. So it's very important to take into consideration the type of tissue that you're homogenizing, because they don't all have the same requirements. For example, while soft tissue like brain or spinal cord can easily be homogenized using simple bead-based methods, other tissues like liver or kidney or gut tissue may need a second round of homogenization to ensure proper homogenization step has been achieved. So it's very important to consider those parameters early on in method development to ensure complete homogenization of study samples. We've also had examples, which I will describe later on in one of the case studies, in which the stability of the oligonucleotides in one tissue matrix may not be the same as in other ones. And so it's very important to consider that not each tissue will be behaving the same, and they may each have their own characteristics that will influence the stability of the oligonucleotide in question. Failure to fully homogenize the samples may actually cause the oligonucleotides to be trapped in tissues, and that will obviously result in under-recovery of the oligonucleotide concentration in that sample. So So now I would like to move on and discuss 3 case studies that we have worked on at Altasciences, which illustrate each of the 3 challenges that I've shown you earlier in the previous slides. So in this first example, I would like to talk to you about a case study in which the client came to us with the desire to redesign a method from scratch in order to have a better sensitivity than the one that they had previously from a third party, using the liquid chromatography with fluorescence detection system. The sensitivity that they had achieved was 10 nanograms per ml, and so because of that, all the samples that they had analyzed with that method were all BLQ. And so our objective was to try to reduce that by at least 10 to 20-fold and try to get concentrations for all these samples. So the approach that we took was to design different probes and then compare those different probes with 2 different assay formats, the dual hybridization as well as the hybridization ligation assay. And we've also used different platforms, fluorescence and ECL. The goal was to get the best sensitivity. As I mentioned, we needed to lower it, but also to pay attention to interferences in terms of metabolite cross-reactivity and ensure that we don't have or minimal cross-reactivity with metabolites. So the first set of results that we got with the fluorescent platform is that the LLOQ that we were able to achieve was about 1 nanogram per ml, which is, in comparison with the previous method that was developed by a third party, was still better. However, our sponsor was looking for even lower sensitivity. And so the approach that we took for this project was to try to test the ECL platform to increase the dynamic range. So the next step was to test the hybridization ligation assay as well as the dual hybridization assay using the ECL platform. And in this experiment, we've looked at N-1 and N-2 metabolites in both to see whether one is better than the other platform in terms of cross-reactivity. And not to our surprise, actually, we found, and consistently with literature, we found that, the hybridization ligation assay has much lower cross-reactivity when compared to the dual hybridization assay. And so moving forward, we decided to continue with the hybridization ligation assay. So then we looked at the precision and accuracy of the method and whether we are able to go to 0.5 nanograms per ml to really increase the sensitivity of our assay. And in fact, the results that we achieved in precision and accuracy were showing that the 0.5 nanograms per ml is something that we can achieve consistently and reproducibly. So in this second case study, I'd like to discuss an example in which we had issues with matrix effect. And so in this study, we were developing a method to support the concentration determination of oligonucleotides in human feces homogeneous. And so what we found is that the blank had a very high signal, which was impacting selectivity. And so there's a difference in signal between lots was so high, up to, in fact, fivefold, that the selectivity evaluation was not able to meet the acceptance criteria of 80%. And so there were different types of hypotheses that were suggested. The first one was that the matrix effect was due to the nature of the matrix in question, which is species. And the second one is around the hybridization conditions that were used in this assay. And so what we did to eliminate the variability between the lots is that we used a larger pool of QCs and standards. So instead of using 4 lots to make up the standards and QCs, we used 12 different lots. And so when we did that and we tested the different lots, separate individual lots that are shown here, we found that the selectivity still did not meet the acceptance criteria. However, we were able to see an improvement compared to previous results. So after having increased the number of lots that we used for creating the standards and QC, we also started playing with the parameters of the assay itself. So one of the things we did is to increase the MRD. By doing that, the signal itself was compromised, and therefore we followed with increasing the LLOQ of the method. And so by doing all 3 steps, we were able to achieve selectivity, and those sets of parameters were then used to validate the method in question. And so the take-home message is that each matrix will come with its own characteristics, and so selectivity may be encountered with one type of matrix that is not for the other. And so it's very important to test and evaluate those for every matrix that you're looking into. And finally, the last case study that I'd like to present is one in which we had issues with stability of the oligonucleotides, a specific type of matrix. In this particular case, we had several projects to support a specific oligonucleotides in plasma for preclinical studies in monkeys and rats, and those were very successful assays and methods that were used. Our objective was to transfer the previously developed bioanalytical method into human gut tissue. And so for that purpose, we had developed an ECL method that was transferred from monkey plasma to human gut tissue. In that process, all critical evaluations for selectivity, specificity, precision and accuracy, dilution linearity, and prozone were successfully obtained. However, the issues that we were having is around the stability of the oligonucleotides in human gut tissue at the homogenization stage. So on this slide, I show you the results of the freeze-thaw and short-term stability of the oligonucleotide in human gut tissue. You can see that following 3 freeze-thaw cycles, the oligonucleotide inhomogenate is not stable, as there's no, as it's under recovering. We've tried to use proteinase K in order to see whether that would limit the degradation of the oligonucleotides. However, as you can see, there were no differences whether we add or do not add the proteinase K. We obtained also similar results when we looked at the short-term stability with or without proteinase K, indicating that it's really a question of stability of the analyte in homogenate and not only due to the freeze-thaw cycles themselves. So this is just to show you that an example in which an oligonucleotide that was providing very good stability in plasma and other types of tissues that we had analyzed when conducting preclinical studies did not show the same situation here, and how important it is to evaluate those types of stability in each matrix that you're working on. And so what we try to do is to ensure that at least the oligonucleotide is inhomogeneous, is stable for at least 4 hours, which will allow us to perform the extraction right after the homogenization stage so that we can limit any type of oligonucleotide degradation. And so the table that you see here shows that whether at 1 hour, 2 hours, 3 hours, or even 4 hours, the oligonucleotide is stable. As you can see, a good recovery up to 4 hours. And so that provided us a comfort that the oligonucleotide in homogeneous gut tissue is stable for at least 4 hours. And so in conclusion, we can just mention that the gut tissue homogenization is not stable for anything longer than 4 hours. Therefore, what we intended to do for this study was to perform the extraction right after the homogenization stage. And so when we also looked at the extraction freeze-thaw, short-term and long-term stability, and those were actually acceptable. They met all acceptance criteria, and therefore we moved forward to validating this method. So again, looking into different matrices, what's important is to really evaluate, we call it pre-validation, in which you really look at different parameters for the assay performance, selectivity, stability, precision and accuracy, prozone, to really get the sense of how the method is performing before starting validation, in order to react and fix the method before encountering any of these issues during validation. And finally, to conclude this webinar, I'd like to just go over some key take-home messages. The first one is around ensuring that all types of interferences are being evaluated in the method that is being developed. Second point, oligonucleotide concentration requires high level of sensitivity in order to achieve the accurate PK profile at the preclinical stage, which can drive different decisions. And so it is very important to try to attain a good level of sensitivity. And then finally, since there are no ICH or FDA regulatory guidelines for validation of oligonucleotides, our strategy is to follow the general principles outlined in the FDA 2018 as a starting point for method validation where applicable. And with that, I conclude my webinar, and I will take on any questions from the audience. And now I'd like to take the different questions from the audience.
Thank you for that really insightful presentation, Danielle. I hope our audience found it of great interest and value. If you do have any questions, then please continue to submit them using the Q&A tool in the right-hand side of your screen. We've received a number of questions so far, so we'll begin to address some of these now live on the air. So the first question for now, Danielle, is why does the Ligation Hybridization ELISA have less metabolite cross-reactivity when compared to dual hybridization ELISA?
That's an interesting question. Yes, well, the answer is really due to the ligase itself. So for the ligation to occur, we need to have the analyte that is in a very close proximity with the ligation probe, and that proximity will allow the ligation to be effective and to occur. If there's any gaps due to shorter lengths of the analytes, i.e. the metabolites, then the ligation step cannot occur between the analyte and the probe, and therefore the oligo itself cannot be quantified.
Thank you very much. And we have another question here, and this one is as follows. What challenges have you encountered when shifting to ECL platforms for the hybridization assays?
Actually, we found that the ECL platform are much easier and much more convenient to use and much shorter in length than the ELISA platform. Of course, as I discussed in my presentation, what comes with ECL platform is a much lower sensitivity, and therefore what's important is to ensure that there's a good separation with the blank. And so this comes with additional levels of optimization that is required to ensure that that you can detect a good separation there. But otherwise, we much prefer actually the ECL platform.
Thank you very much. And we have another question here. So the third question is, have you encountered any issues with ADA interferences in your hybridization ELISA assays?
Actually, no. The proteinase K or the high level of temperatures are able to degrade all types of proteins, including ADA. So but we still need to test for it in our validations, and so far we haven't seen any interference in the assay, in any assay that we've developed and validated.
Thank you very much. And another question that we've received is, when homogenizing tissue samples, what precautions do you take to ensure the integrity of each sample?
Yeah, thank you for that question, because when it comes to tissues, it is more finicky because you do have to use a polytron to homogenize the tissues, and to ensure the integrity of the samples that you're testing, we have a very, very rigid procedure that ensures a good washing steps is included between each sample. And then also at the end of each day, the whole polytron is disassembled, all the blades are disassembled and washed and cleaned before the next set of samples are tested the next day. So it's a very lengthy procedure which takes quite a long time. So, and all this to ensure that there's a good integrity around our tissue samples.
Thank you very much, and we've got enough time for one more question. So the last question for today is going to be, how do you explain the lack of stability of oligonucleotides in gut tissue homogenate?
That's a good question, and we did think a lot about this when we were trying to resolve the stability of the oligonucleotide in gut tissue homogenate. So what we suspect, and of course we don't have any evidence for that, is that it's probably due to bacteria in the gut tissue that would have some nucleases. And once you homogenize, those nucleases are being released and may act on the oligos and degrade those oligos. And so, that, of course, there's no evidence for that. It's just what we suggest could be the mechanisms involved.
Thank you very much, and I'm afraid that is all we have time for today, so we will address all of the other questions offline. If you do have any other questions, please feel free to send them in to webinars@bioanalysis-zone.com. I would like to once again thank our wonderful presenter, Danielle, as well as you, our listeners, for your time and your questions. If you would like to let us know your thoughts on today's webinar, please feel free to complete our feedback survey in the resource widget or tweet us at BioAnalysisZone. Don't forget to visit us at www.bioanalysis-zone.com for more webinars. And thank you again for attending, and goodbye.